Yes.
Okay, recording is in progress. So
I'm going to show this to do a short introduction, okay? And yeah, so welcome everyone to this 15th webinar, ECI Webinar of Translacore. And today we have Jesús de la Cruz and Sara Martín-Villanueva. Jesús de la Cruz is a PI of Synthesis and Function of Ribosome Lab at the Institute of Biomedicine in Seville, in Spain. And Sara Martín-Villanueva is a postdoc in Jesús de la Cruz Díaz lab. Okay, today she's going to talk about decoding AL22, how a non-essential protein becomes critical for ribosome maturation. So yeah. You can start now. Thank you so much.
Well, we will share.
No,
So here
share,
now.
share.
Yeah.
No. Aquí.
Share.
Desktop 2.
Desktop 2.
Share.
Share. And
now you have
PowerPoint, no?
Yeah.
So
Like this.
Do you see my presentation?
Yes.
Yes.
Now we.
And what is the license point? Yeah. Okay, stop. So thank you very much. So I am supposed to give an introduction of what we're doing in the lab, and then Sara will give her seminar. So the first things I would like to do is just to thank Cecilia, no, and the rest of organizers, no, to give us the opportunity to speak, to present our work, no, in this webinar. And obviously all the members of the Translacore course that are connected to hear us. No, as Cecilia said, our lab is called Synthesis and Function of Ribosomes, and we work at the Institute of Biomedicine of Seville. That is a building that is our official web page. Say that we promote, our center promote fundamental research, with the aim of immediately transfer that research to knowledge, to the clinical reality and the development of new therapies. And as Cecilia has also said, our institute is in the beautiful city of Seville. Although the weather in summer is too hot, but well, nobody is perfect, We work in the subject of ribosome biogenesis in eukaryotes and in translation, but our aims specialize in the functional characterization. We use a lot of genetics in our research in order to understand the role of ribosomal proteins and ribosome assembly factors in these two processes, ribosome biogenesis and in translation in eukaryotes. And our favorite organism, due to the versatility, is the yeast, Saccharomyces cerevisiae, although we also work in some human cell lines. As everybody in this course knows, ribosome biogenesis is a very, very dynamic process that spend a lot of practically the majority of the energy resources of the cells. It's a very dynamic process because about 2,000 ribosomes are done each minute in a rapidly growing yeast cells. So the process starts with the transcription of the precursors of the ribosomal RNA. So you have, as all of you know, you have a lot of process. So these precursors get modified, folded, and processed, and at the same time, concomitantly to those processes, ribosomal proteins assemble. By the help of the myriad of factors known as transacting factors or ribosome assembly factors. And the particles mature, get exported, mature a little bit more in the cytoplasm. Sara will speak about that today. And and the ribosomal subunit become functional and translate in the happy end in the cytoplasm. This is this picture that was given by Ivo, friend of mine in the Kutai lab, showed basically that dynamic. That as soon as the precursors of ribosomal RNA are transcribed, the processes of modification and processing of ribosomal RNA occurs inside ribosomal pre-ribosomal particles. That these ribosomal particles mature first of all in the nucleolus, then in the nucleoplasm, and finally in the cytoplasm. And apparently, the maturation of the large ribosomal subunit and the small ribosomal subunit are independent of each other. Regarding the large ribosomal subunit, there are so many experimental evidences that they say that the maturation occurs in a kind of from the end to the middle model. First of all, the ribosomal RNA that is divided in 6 domains, from domain 1 to domain 6. The domain 1 and domain 2 are apparently the first ones to be assembled, to be folded with the help of a lot of ribosomal assembly factors. Some ribosomal proteins from those domains assemble, and together, and this after assembly of domain 1 and domain 2, part of the domain 6 also join to the ribosomal subunit information. Then domain 5, the rest of the domain 6, domain 4 and domain 3, together with the 5s RMP are associated, and 5s rotated, and finally the mature ribosomal subunit is done. And as I said, one of so the interest of our lab for decades, one of those aims is to study the role of during assembly of a specific ribosomal proteins. And from here to the end of my introduction, what I did just was an overview of all ribosomal proteins we have studied. Starting from ribosomal proteins from domain 1, we have studied the role of L15, L36, and L8 during the shaping of domain 1, and so that we conclude that these ribosomal proteins are required for proper pre-processing of the 27S pre-ribosomal RNA. So, and the assembly of these proteins to the pre-ribosomal particles is essential for the association of a set of transacting factors called belonging to the family of A3 and B factors. And furthermore, our data suggests that the assembly of these 3 ribosomal proteins, L8, L15, and L33, occurs hierarchical, in a hierarchy. So meaning that the assembly of L8 is happening before that of L15, and then L13 is the last one binding. We have also worked in these 2 proteins, L14 and L16, from domain 2, that are also very assembly proteins from domain 2. And in this case, again, these 2 ribosomal proteins are required for proper processing of 27S pre-ribosomal RNA. And these 2 proteins, first of all, are required for the association of the complete set of the factors belonging to the A3 family. And the assembly, we also demonstrate that the assembly of L14 and L16, that is called UL13, is required for assembly of the rest of the ribosomal protein belonging to domain 2, between them L20, L33, or L6. So we have worked a lot together with John Woolford in the functional characterization of a set of ribosomal proteins, L22, L24, L26, L35, L37, L39, that are proteins that the feature of these proteins is that they surrounded the what is called the exit part, so the cytoplasmic part of the tunnel of the ribosomal of the peptide exit tunnel. And we have worked for a lot of time with these proteins. And we show that these proteins, so the assembly of this protein need the previous association of the A3 factors that I have speak before, and then the assembly of these factors of this protein, is required for the further binding of the B factors, especially those factors called NSA2 and NO2. So meaning that if this assembly is done, processing of the proximal stem is occurring, and if the assemble do not occur, for instance, upon depletion of any of these members, so the NSA2 and NOC2 factors do not get associated into the pre-ribosomal particles, and then those particles are rapidly turned over. So our favorite ribosomal protein is UL3. Okay, we have studied two different functions of UL3 during assembly and translation. The first function of L3 that we know that we have studied is the function of L3 in assembly, that is related to the function of the complex called the DBP6 complex of MPA1 complex. L3 have the feature, it's a very special protein because L3 binds to all the roots helices of all domains, and also binds to the boundary between the double helix formed by base pairing of the 5' end of 5.8S ribosomal RNA and the 3' end of the 25S ribosomal RNA. And also together with Simone de Baron, that is in the audience, and together, so in collaboration with the David Tolleby group, we found a very nice function of L3 regulating the function of FAN12, that is called also EIF5B, during the maturation of the small ribosomal subunit. So basically the 3' end of 18S ribosomal RNA processing during the cytoplasmic maturation of the ATS-like particles. And then finally, we have been also working with two proteins, L40 and P0, that both proteins have the particularity that the assembled during the cytoplasm are required for the proper function of some transacting factors that are working in the cytoplasm, and that's it. The protein we are working now in the lab is L22. Then Sara will present the data we have obtained in the characterization of L22 related to the cytoplasmic assembly steps of the large ribosomal subunit, especially L22 have something to do with ARX1 and RA1 factors. But I will not obviously spoil the seminar of Sara. Would like to thank the people in the lab involved or not in the group. Obviously, all the collaborators have been working with all these ribosomal proteins, and that's it. And by this short or long presentation, I have done an overview of what we are doing with ribosomal proteins. And now it's time for Sara. I would like to thank all of you for your attention, and obviously I would like to thank Sara for her willingness of giving this webinar. Okay. Thank you very much. And I leave Sara. I will leave the room and join the rest of the group to leave her more quiet. Okay. Thanks a lot. And
Thank you.
if you have questions, better to have all questions at the end of the
Yes.
webinar. Okay. Thanks a lot.
Thank you. Thank you so much.
One moment, because I have to change. Can you see my presentation? Yes. Okay. Thank you. So I'm Sara, and first of all, I would like to thank Cecilia and the organizing committee for giving me the opportunity to share our work with you. And as Jesús said, today I'm going to talk about the study of the ribosomal protein L22 and his role in ribosome maturation. And why L22? So L22 is a non-essential globular protein. Non-essential globular protein, and is a eukaryotic-specific protein. And although it has been determined that it's assembled in the nucleolus, his role in ribosome biogenesis is still unknown. L22 is located close to the peptide S-cell tunnel, and moreover, in cytoplasmic particle, L22 interacts with the transacting factor RA1, and that is a factor that verifies the correct formation of the peptide S-cell tunnel. And as Jesús said, our group is particularly interested in the polypeptide S-cell tunnel, and we have several studies in which we have characterized the proteins that are close to the tunnel. And finally, human L22 is the second ribosomal protein more misregulated than in cancer, although it's not known why. So we started studying the timing of assembly of L22, and to do this, we use a system in which in galactose medium, the nuclear cytoplasmic export of pre-60S particle is blocked. We analyze the localization of L22 touched with GFP, and while in glucose medium, the signal is cytoplasmic, in a medium containing galactose, L22 accumulates in the nucleolus, suggesting that L22 associates with pre-60S ribosomal particles already in the nucleus. To better determine the timing of assembly of L22, we next purify particles containing L22 GFP and determine which pre-ribosomal RNA will purify with them. As we see by ordered blot, L22 was not able to purify the pre-ribosomal RNA 35S and 27SA, but is able to purify the precursor 27SB and the subsequent intermediates like 7S. This means that if we look at the ribosomal RNA processing scheme, L22 is not present in early particles, but is present in later nucleolar particles. So we can conclude that L22 stably associated with middle nucleolar pre-60S particle, but not at very early stages. We also study the phenotypic characterization of L22. In yeast, L22 is encoded by two paralogous genes, RPL22A and RPL22B, which produce practically identical proteins. We analyzed the phenotypic consequence of growth of deleting both genes, and we observed that cells lacking the B copy display a wild-type phenotype at all the temperature that we have tested, but on the contrary, deletion of the E copy display result in a severe slow growth phenotype, especially at low temperature, at 22 degrees. And the phenotype of the double mutant is the same that the deletion of the B copy, meaning that the responsible of this growth is this copy, the E copy. So this result indicates that L22 is required for optimal growth, especially at low temperatures. Well, as we are interested in to study the role of L22 in ribosome biogenesis, we analyze the translational machinery status of the double L22 mutant by polysome profile analysis. On this kind of profile, the first bit corresponds with free 40S ribosomal subunit, the second one with free 40S ribosomal subunit, and the third bit are in specific joining of both subunit or messenger RNA molecule with a ribosome engaged in translation. And the rest of the bit correspond to messenger RNA with 2, 3, 4 or more ribosomes engaged in translation. And compared with the wild-type strain, the double mutant L22 show a decrease in the 60S peak, and we also observe this peak that we call Halmer, that corresponds with a messenger RNA molecule with a ribosome engaged in translation, but also with the small ribosomal subunit waiting for the large one. This kind of peak are usually typical of joining defect or 60S deficit. So this result suggests that absence of L22 results in a deficit in 60S ribosomal subunit. On the other hand, when we analyze this profile at 22 degrees at 37 degrees, we observe that at 22 degrees, the polysome profile is very similar to that at 30 degrees, while at 37 degrees, this phenotype was also completely suppressed, indicating that the defect in subunit production is also dependent of the temperature. Next, to determine if the 60S defects observed in absence of L22 could be due to an impairment of the pre-ribosomal RNA processing, we assess the steady-state level of pre- and mature ribosomal RNA. And we observe in the double mutant L22 an accumulation of the precursor 35S and 27S. And also the mature 25L is mildly decreased. To resolve with more precision the different form of 27S pre-ribosomal RNAs, we perform primer extension analysis, and we observe that both types of 27SA don't vary the levels, but however, both forms of 27SB accumulate in absence of L22. Furthermore, consistent with the growth analysis, these defects in pre-ribosomal RNA processing are more drastic at 22 degrees and almost completely suppressed at 37 degrees. So all these results suggest that L22 is necessary for normal processing of the pre-ribosomal RNA, especially for the processing of the precursor 27SP. Next, to further evaluate the consequence of the absence of L22 on 60S maturation, we determine if the nuclear export of pre-60S ribosomal particle was impaired in the double L22 mutant. To this end, we analyze the subcellular localization of a GFP-touch version of the reporter L23, which assembled in the nucleus, and in a wild-type strain, the steady-state level of the protein was cytoplasmic. However, in absence of L22, we observe the signal was distributed between the cytoplasm and in the nucleus, suggesting that L22 is required for the efficient cytoplasmic export of pre-60S particle. And this is specific of the last ribosomal subunit, because if we analyze the localization of a reporter of the small ribosomal subunit, we cannot find difference between the wild type and the mutant L22. As a summary, L22 is important for ribosome maturation, and in absence of L22, we have an accumulation of 27Sb pre-ribosomal RNA and a nuclear retention of pre-60S particles. Interestingly, depletion of some essential ribosomal protein that contact with domain 3 also have the same phenotype. That are the protein that I have marked here with a red star. So, despite L22 is not an essential protein, and this suggests that L22 has an important role in the assembly of the ribosomal RNA domain 3. Next, once analyzed the main phenotype of absence of L22 in ribosome biogenesis, we wanted to study the relation between L22 and some important ribosome biogenesis factor. In the structure of the cytoplasmic pre-60S particle, L22 is located close to the peptide acyl tunnel and is close to the transacting factor RE1. This transacting factor proves the correct formation of the peptide acyl tunnel, and as we can see here, have a C-terminal tail that fully enters in the acyl tunnel. And moreover, L22 directly interact with RE1, with the initial part of this C-terminal extension. For this reason, we decided to investigate if absence of L22 impact of the role of RE1. Interestingly, similar to absence of L22, lack of the assemble factor RE1 also induce a slow growth phenotype, especially at low temperatures. And we constructed a double L22 RE1 mutant, and we observed an enhancement of the growth defect, especially at low temperatures, suggesting that these two proteins are linked. And during ribosome biogenesis, RE1 work together with the ATPase SSAA and the J protein JJJ1. So we decided to analyze also the relation between L22 and the J protein JJJ1. And to do that, we introduced a JJJ1 deletion into the L22 mutant strain, and we observed that loss of JJJ1 only very slightly enhanced the growth defect of L22 null cell. And moreover, if we also analyze the effect of the overexpression of these two factors in a delta and a double L22 mutant, and we observed that only the overexpression of RE1 partially suppress the growth defect of the L22 mutant strain, suggesting that maybe L22 could modulate the association or function of RE1. And what is the function of RE1? RE1 is necessary to remove the assemble factor AX1 and ALK1 from pre-60S particles. These two factors, ARS1 and ALK1, are also assembled very close to L22, and these two factors are thought to work together and redundantly as a heterodimer, and they are also important for the functional check of the peptide acyl tunnel. So we also analyze the functional link between L22 and these two factors. And we introduce a mutation of these two factors into the L22 null strain and analyze the growth. And we found a very interesting phenotype because deletion of both genes suppress partially the growth defect of the L22 mutant strain, especially for the deletion of ALK1. So for cells, it's better not to have L22 and ALK1 or AX1 that only don't have L22. But why? This genetic interaction suggests that defects in the L22 mutant are caused, at least in part, by the presence of these two factors. So to test this hypothesis, we wonder if the deletion of ALK1 also suppresses the other phenotype of the L22 strain. We wanted to know if suppressed at ribosome biogenesis level. we focus on AL1 because the effect of the suppression was more evident than for AS1. And so we analyze if deletion of AL1 is able to suppress the 60S deficit of the double mutant by Poisson profile analysis, and we observe that loss of AL1 partially suppressed the deficit in 60S ribosomal subunit. To better see this suppression, we quantify total ribosomal subunit by low magnesium sucrose gradient, and if we look the 60S versus 40S ratio, we observe that the double mutant suppress the deficit in 60S ribosomal subunit. We have in the double mutant a ratio of 1.43 versus the 1.50 ratio of the L22 mutant. But this ratio is not as high as a wild-type strain. So we can conclude that loss of AL1 partially suppressed the deficit in 60S ribosomal subunit nude cells. Given that for cells it's better not to have L22 and AL1 that only don't have L22, we analyze if the recycling of this factor is affected in absence of L22. In a wild-type situation, some ribosomal transacting factor like ARS1 and ALP1 bind to pre-60S particles in the nucleus, and they are transported to the cytoplasm with the particle. This shedding factor must be removed and recycled back again into the nucleus to participate in a new round of biogenesis. So in a wild-type situation, some of these factors like ARS1 and AL1 localize in the nucleus by fluorescent microscopy. But what about L22? Is the release or the nuclear import of this factor affected in absence of L22? The answer is yes. We observe a mislocalization of R2 and AL1 in the cytoplasm in L22 null mutant cells. So this suggests that this protein L22 is important for the recycling of this factor. As a control, we also analyze the cellular distribution of the shedding factor NRT4, which localizes far away from L22 and is involved in a different branch of the cytoplasmic maturation of 60S. And in this case, we cannot find difference between the wild type and the L22 mutant. And we also analyze the cellular distribution of the transacting factor NOF1 that is involved in initial cytoplasmic release event, and in that case we cannot find difference. So the recycling of these two factors seems not to be impaired in absence of L22. And finally, we analyze if absence of L22 affect the recycling of disease of the yeast homolog of human EIF6, another assemble factor that is released after AS1 and AL1, and that it's essential in ribosome biogenesis because it prevents subunit association. And while in a wild-type strain, TIP6 is mainly observed in the nucleus, in absence of L22, TIP6 GFP was mislocalized in the cytoplasm. So the recycling of disease seems also to be affected in absence of L22. So according to the current hypothesis in the field, RAY1 together with the ATPase SSA and the J protein JJJ1 is important for the release of the transacting factor AS1 and AL1. So in absence of RAY1, these two factors cannot be properly released, and the presence of these two factors in pre-60S particle avoid the progression of the cytoplasmic pre-60S ribosomal maturation. So we propose that in absence of L22, RAY1 cannot perform his function or is not properly recruited. So these two factors, AS1 and AL1, persist in pre-60S particle that cannot mature properly. It will be like a checkpoint that monitor the progression and integrity of 60S ribosomal maturation. So we wonder if the defect observed in the L22 mutant were due to the persistence of these two factors on cytoplasmic pre-60S ribosomal particles. Interestingly, it has been suggested that large portion of the ribosomal protein L25 blocks ARS1 binding to 60S ribosomal subunit. Because these two proteins binds close in the particle. And we found that if we introduce a plasmid that expresses this protein tag with GSP, we observe a partial suppression of the growth defect of the null strain. And moreover, we also study a mutant of ARS1 that is defective in 60S binding, the ST347P mutant. And we observe that this mutant also suppresses the growth defect in the L22 mutant strain. So this result suggests that the defect observed in the L22 mutant were due to the persistence of at least the transacting factor ARS1. And again, according with our hypothesis, we also propose that in absence of L22, RA1 cannot perform his function, or maybe is not properly recruited to pre-60S particles. So we want to analyze if the recruitment of RA1 is affected. To address this question, we separate extract from L22 and wild-type strain of sucrose gradient and analyze the distribution of this factor by Western blot. And we observe that RA1 is present in pre-60S particles in both wild-type and L22 mutant cells, suggesting that absence of L22 does not impede the association of RA1 to pre-60S particles. And moreover, in consonance with our hypothesis, ARS1 is also present in pre-60S particles in both strains. And to further analyze the association of RA1, we purify pre-60S particles containing ARS1 by immunoprecipitation. And by Western blot, we observe that in absence of L22, RA1 is present in pre-60S particles to the same extent as in the wild-type strain, confirming that absence of L22 does not impede the association of RA1 to pre-60S particles. So as a conclusion, we propose the next working model. In a wild-type situation, L22 associates with pre-60S particles during middle nucleolar stages of assembly. Ribosome biogenesis continues, and some ribosomal transacting factors like ARS1 and AL1 bind to pre-60S particles in the nucleus and are transported with the particles to the cytoplasm. These shutting factors must be removed and recycled back again into the nucleus to start a new round of biogenesis. But according with the current hypothesis in the field, RA1 is important for the release of these two factors of AL1 and AL1. So in absence of RA1, the recycling of these two factors is impaired, and also is impaired the progression of the cytoplasmic pre-60S ribosome particle, which include the release of TXCs. Our results suggest that lack of L22 result in 60S maturation defect, like deficit in 60S ribosomal subunit and defect in pre-ribosomal RNA processing. Moreover, in absence of L22, some factors like NOH1 and MRT4 are properly recycled, but other factors like ARS1 and AL1 and also TXCs accumulate in the cytosol, suggesting that L22 is important for the recycling of this factor. And given the similarity in phenotype between L22 and RA1, our result also suggests that L22 may modulate the function of RA1 during cytoplasmic maturation of the 60S ribosomal particles. Just to finish, we are also analyzed the role of L22 in ribosome biogenesis, but what about ribosome function? L22 is not an essential protein, so ribosome without this ribosomal protein can translate. So here we have a homogeneous system with aberrant ribosome. So we want to know how this ribosome translates. And to know this, we have done polysom profile experiment to compare the total translatom of the wild-type strain and the L22 mutant. Unfortunately, we are still analyzing the data, but in any case, to finish, I want to point out that with this technique, we can obtain also a lot of information. For example, we can know the initiation rate of the translation efficiency in this mutant. So I want to thank all members of the lab, especially the people involved in this project, Jesús de la Cruz, José Fernández, Carla, Cristina, Paula, and obviously I would like to thank all the institutions that support our project, our main collaborator. Thank you.
Thank you, Sara, for this amazing talk. Really, really nice. So now it's time for some questions. So if you want, you can ask some question or say something to Sara and Jesús. If you want, I can start. So really nice presentation. So I was wondering if you have checked the viability or the longevity of this mutant, because I was checking this mutant in PubMed, and I checked that has a role in replicative lifespan, right? So I mean, if you have checked some of the viability of the longevity in this mutant and these double mutants that you did.
We have not done this, but we know that L22 is also important for other functions, like for example for longevity and also for meiosis, and has another important function, but we only focus on ribosome biogenesis.
Nice. Okay, thank you. Thank you so much.
Let's see what can we get from the ribosec.
Yeah.
It's also an important point. So you know,
We'll have a lot of information there. Yeah. Thank you. So now Anthony first.
Hello, everybody. Thank you, Sara. It was a very nice presentation. Hello, Jesús. I just have a small question on the recruitment of L22. You are excluding a very early recruitment and proposing that it's recruited more in the pre-60S particle containing the 27SB precursors, but both on your IP experiment, there are little amounts of 35S, which is co-purified. It's not a lot, but still. And when you deplete L22, you have 35S accumulation defect. So I would conclude from that the recruitment is more early than what you propose. So can you comment on that?
Yes, maybe it's true. We cannot discard an earlier association of L22. And another thing that we want to do is to purify particles and see the composition of these particles, if to know
Yes.
if L22 is present earlier. But we don't have the results yet.
So that the point is that properly, maybe we didn't use the proper term. You know, the stably assembly of L22 occurred late. Obviously, we don't know if assembly is occurring earlier, because you know the association, as Philippe Milqueret described nicely, you know, in papers a few years ago. So first of all, you have the assembly that is not quite stable, and then you have when RNA is properly fold is when you get the stable assembly. That is the thing we can see after IP. But obviously, we agree with you. So many proteins described to assemble at middle stage of the assembly pathway are associating earlier with the pre-processing modeling.
Thank you.
Thank you, Anthony.
Thank you so much. And now it's Julie. Julie, if you, yeah.
Thanks. A lovely talk, Sarah. I wanted a little bit more about like the temperature phenotype that you were seeing for the A paralog, and whether you, when you're doing the RiboSeq, are you doing that at 37, or are you trying also a different 22 to see if to get a handle on what's going on at there?
And we have done the ribosome profiling at 30 degrees at the normal temperature, but we don't discard to repeat the experiment at a different temperature because I think we can see different things at a different temperature.
Yeah, with it. Well, you can. This is a very, very nice comment. So the 30 degrees, so 30 degrees, let's say the standard temperature. So we would like to to have very clear the results at 30 degrees and then check particular genes at the 22 degrees. We are hesitating to use 37 because at 37 degrees we don't have a phenotype, or we don't have growth defect, and perhaps this is due to the that the high temperatures ARX1 and that is not an essential factor. Due to the property of the high temperature, is dissociating spontaneously more oftenly, and that's why the role of L22 is not as critical as at low temperatures. So we don't expect, but this is a very nice control. So we will do the experiments at 30 degrees and then check those particular genes being differentially expressed at the lower temperature, and maybe after your comment also, why not at 37 degrees?
No, sorry, that was my naivety, was the 37. I forget that yeast grow at 30, sorry. But it was more the lower temperature, because I was trying to think about the fact that it's near the exit tunnel and potentially maybe the contribution is something to do with temperature has such a big influence on folding proteins, and whether that's the point at which that sort of temperature sensitivity has come into place. So thanks.
So far, although this is a pure speculation, so far we understand the growth defect of L22 regarding the temperature as a kinetical problem of ARX1, although we don't have any results saying that the ARX1 is releasing easily at 37 degrees and less easily at 30 degrees and hardly at 22 degrees. But
Thank you.
Thank you.
Thank you. Now it's to Alan. Yeah.
Hi. So as you know, the binding of REI1 is quite complex. So there's the C-terminus is in the exit tunnel, and the zinc fingers bind to the subunit face and interact probably with the L22. So I just wondered how you thought. So even though you see REI1 is still bound, it may not be binding in the normal way. So maybe you lose the contact with the subunit face, or maybe you lose insertion into the tunnel. I just wondered what you thought about that in the absence of L22.
Yes, you know that you will be the one solving that question. Hopefully, our idea is to together with your expertise to prepare particles after purifying ARX1 and send it to your nice lab, just in order to perform some cryo-IM and check how RI1 is associating in the absence of L22. So what we believe is that as the middle part of RI1 is interacting with L22, so when you don't have L22, so the interaction should be somehow compromised, meaning that perhaps, but this is all again pure speculation, perhaps the association with the JJJ1 and SSA 1, 2, 3, 4 is not occurring as properly as in the wild type situation, and this is the consequence the delay, because indeed it's a delay. L22 is not essential. ARX1 is not essential. RA1 is not essential. So in the absence of those factors of ribosomal protein, what you have is a delay in releasing the factors. But we hope to give you samples before spring.
We look forward to receiving them.
Thank you.
Thank you so much. And now, Simon, you want.
Yeah, Sarah, thank you. Nice talk. Again, going back on these previous comments. So on your next experiment on the ribosome sec to analyze the translatum effect, are you also planning to use this double mutant LPL22 and ARX1 or LB1 mutant to compare that? Although you recover the growth, you might also go through a different translatum. So it would be also interesting to see if you completely fully recover the same translatum than wild type, or I don't know, if you.
Wonderful. As I said.
Yes. Thank you, because it's a good idea. But as Jesús said, now we only start with the double mutant and only with one temperature. But after that, I think it will be very interesting to know what happened with a mutant without L22, but with a normal growth.
This is as Sara said, this is a very wonderful comment. Again, as I tell to Julie, our idea is to get clear the translaptome of the mutant along 30 degrees, and then starting to check for particular functions, because we started to suspect that they are, group, the ontology group of factors related to some functions affected, differentially affected. And the idea is to check those factors or the consequences of the lack of a proper translation of those factors in different circumstances. So, for instance, different temperatures of L22, and also if we can suppress, as you nicely comment, if we can suppress the defect, the translational defect, once we suppress the growth defect of L22. Yeah. Wonderful question.
Celia, if you want.
Yeah, sorry. Thank you, Sarah. It was a wonderful talk. I just have a comment more than a question. We work on mouse cells that have been depleted of L22. So we have a homozygous mutant of L22 in mice cells. And on those ones, we performed the ribo-seq experiments, and we don't see any translation efficiency differences between wild type and mutants. So, but also on the ribosome gradient profiles, we don't see any differences. So you might be on something very interesting, because it's in terms of evolution, there seems to be something different. So I just wanted to tell you that. And also I was wondering if you had thought about when you look at where L22 is on the ribosome, so it's close to the peptide tunnel exit, for sure, and it might indeed play a role in recycling those ARX1, ALB1 factors. But I was wondering whether you thought about extra ribosomal functions of this protein also, because it seems to be, from a structural point of view, it seems to be easy to extract. So do you ever see something like that?
We are not focused on a different function of L22, but one thing that we believe happens is that L22 have more than one function in the ribosome biogenesis. We have a role in the nuclear part and also a role in the cytoplasmic part. But there are also some studies about L22, about other roles in L22 and also in extra ribosomal function of L22.
Yeah. Celia, so obviously we are aware that you and Kim are working with L22, and we discussed on the ribosome meeting, and our idea is just as soon as we have, a complete, although still preliminary, as soon as we have a complete first study of the translocome of this, to share with both of you, just in order to compare. So we were aware of the extra, so that you claim that the role of L22 in cancer must be extra ribosomal, no, in the sense that you don't see any defect, but now I have a point of information different on the one I got in the meeting, because I didn't know that you are deplete cells of L22 do not have a defect at the level of polysome. Is this what you say? No, that you
Yes,
don't.
that's what we saw here. So we have to yes, at the ribosome profile level on sucrose gradient, we don't see any differences. No major differences between wild type and L22 depleted cells. So
And those cells were the one you use for the ribosec.
yes.
Okay.
Yes.
So and why? So the L22 like protein is involved? No, because this good to know, because either in collaboration with you, or so with you and Kim, or ourselves, we were thinking on the possibility of checking whether the depletion of L22 in mammalian cells is also giving the delay recycling of one ALK1, but now that you have that we have the information from your side.
Yeah,
so
we can discuss it later if you want, but we also run northern blot experiments, and we don't see many differences. So it might be really something different between mammalians and yeast. So I don't know.
That's it. Thank you.
Thank you anyway. Thanks.
Thanks to you.
Yeah, if anyone has more questions, I think that.
May I ask one quick question?
Yeah.
So, do you have some more information about how the lack of L22 is affecting the assembling of other ribosomal proteins? Because the 60S is already, we can see based on the polysome profile that they are formed, yes, and how the rest of ribosomal proteins, whether the 60 is fully loaded with all ribosomal proteins or not. Do you have some more information, like, for example, mass spec analysis or something like that?
We don't have the information yet, but we want to explore this. We want to purify some pre-ribosomal particles to analyze the composition of this particle in not only in focus on ribosomal proteins, but also in ribosomal assembly factors.
That's it. That's the idea. So we are collaborating with a group of John Woolford in this particular characterization, and together with John, what we would like to do is to, as Sara said, to purify pre-ribosomal particles in the presence and in the absence of L22, and do that compositional study that you said by eye track, and to see how the lack of assembly of L22 is affecting the assembly of other ribosomal protein, and the association of transacting factors, because we know that L22 interact with RA1, but we also know that L22 in the nucleus is interacting with the NOP1 and with RLP24. So maybe it's that. And we also have data on the as L22 is not essential and we do genetics, we also have double mutants of L22 with other ribosomal proteins from domain 3. And for instance, when we combine L22 with L38, that I don't remember the name in the novel nomenclature. L38 too. So for instance, we get as an enhancement in the growth defect. The same happened with, for instance, with the A copy of L31, that is another protein in domain 3. And we would like to do experiments with L19. Surely that the lack of L22 at the nuclear assembly level is doing more things. Indeed, the 27SB precursors is getting accumulated, meaning that in the nucleus L22 is having a role. It's not only the thing we have seen in the cytoplasm. So we have a dual role of L22, one in the nucleus affecting assembly, and the other one in the cytoplasm interfering or helping the dissociation of FAS1. Thank you very much for the comment.
Okay, thanks a lot.
Thanks so much. And now a question, if you want, Marianne.
Yes, thank you. First question, maybe naive one. If you overexpress ALB1 or ARX1 in the cell, do you expect any phenotypes?
Great question.
Yes, we have done that. I don't know if I have the data. No, I don't have here. No, I don't have here. But if we overexpress this factor, in a L22 mutant, or do you mean in a wild type strain?
In wild type strain, I was thinking maybe you mimic the RPL222 phenotype somehow. No.
In a wild type strain, we don't have any
phenotype.
No, any phenotype.
Do you make it worse when you overexpress it in RPL22 mutant? You make the phenotype even worse?
Yes, exactly.
Okay. Thank you.
It's very subtle, but significant. It's like when we overexpress RA1, that is very subtle, suppressing the growth defect of L22, but it's significant. Yeah, that's it.
Okay, so yeah. Thank you so much, Sara and Jesús, for the amazing talk, and thank you all for the discussion that I think that was really interesting. Thank you for the attention.
Thanks to you. So I have a question. So so we will have the webinar at Friday at 1:00. The rest of them, or so the next one are coming.
The next one, I think that, I don't know, maybe this month, maybe next month, but it's, yeah.
But in December.
In December, right, Adolfo?
Yeah, that's right. But we don't know the exact time, because the time is usually made to fit the speaker more than the community in that sense.
No, because for me, so my general comment is that I'm not too active in this kind of seminars because normally they are at a time that I have other familiar duties to do. And it's a very pity. So most of them I have to see in recorded. But this is not the important point of the seminar is to have it to discuss it. That's why I say that.
Yeah.
And Friday 1:00. Friday 1:00 is a time I can do it. And if the rest of the seminar are at this time, so it will be very nice.
Well, it really depends on the speaker.
Yes.
They decide the day and the time.
That's understandable.
Yeah.
But we will keep it in mind.
Yes.
Yeah, we will suggest it.
Show up the suggestion.
Okay. Thank you so much, and see you